We'll talk a little bit about bacterial genetics and variation. We cannot finish this topic today because it's a long topic with a lot of material. So bacterial genetics. I'm talking about genetics. We are talking about the molecular biology, and we want to know their genetic material, genetic construction, and also variation means that their genetic material, even if it's relatively stable, they can change. They can cause mutation, and that variation or mutation can cause some trouble for us, such as the antibiotic resistance, which the doctors don't like. So we'll try to cover a little bit about this topic. So before you look at this slide, if I ask you the question, what is the genome? What's your answer in your mind? So is the genome equal to chromosome? No. Yeah, I guess I heard your answer. So genome, the genome here is the definition. The genome is all the genetic instructions for some organism, for their development of cellular structure, for their metabolic functions, all the chemical reactions, and also the regulation of the gene function. All these are encoded in the genome. So genome, we are talking about all the genetic material, including the chromosome. But there might be some extra chromosomal DNA, such as in the simple structure like prokaryotic cells in bacteria. It has genome, the big DNA, but it also has plasmid. So if in this cell it has some plasmid, the plasmid is part of its genome, not of the chromosome, but altogether they form the genome. So there's a question for you. What's the genome of human? DNA, chromosome, and the mitochondrial DNA, right? Exactly. So we have 46 chromosomes, so that's part of genome. But in each cell, we also have mitochondria, and the mitochondrial DNA, separate DNA, is also part of human genome. And when people study the evolution of human, it's important to study the sequence of mitochondrial DNA. We can, by sequencing the mitochondrial DNA, we can derive a lot of information. So that's in this eukaryotic cell. The genome includes the chromosomes, and also includes, there might be some extra nucleus DNA, such as the DNA in mitochondria. In the plant, it also includes the DNA in chloroplast. So the chloroplast DNA is also part of this genome. But for viruses, they are more simple, and the viral particle contains only a short genome. Maybe it's a DNA molecule, maybe it's RNA molecule. That's its genome. Even though it's RNA, we still call this the viral genome. Okay? So this is the definition of genome. But what is gene? What's the definition of gene? So here is the traditional definition of gene. Genes are located within the chromosome or plasmid. They are specific regions of the DNA. But for some viruses, the genes are located on the RNA molecule. So a specific region of the chromosome or the plasmid. So here, this part is one gene, this part is another gene, this part is another gene. And they might carry out function in different ways. For gene 1, it can be transcribed into messenger RNA, then translated into protein. So this is a traditional gene. It's a protein encoding gene. But for this one, it encodes not a protein, but a regulatory RNA. We also call this part a gene, even though it does not encode any protein molecule. And this one, it encodes only a transfer RNAs, only RNA, transfer tRNA region. Still, we call this a gene. And also, there might be some region encoding the ribosome RNA. So the ribosome RNA gene. Okay, all these we call them gene. But this is still based on human definition. And if you have a new updated definition, an updated term, and you can include more sequence, more region as gene, such as those part of DNA. Right now we call them junk DNA, okay? Junk DNA. But maybe in the future we found some function in the junk DNA. They might contribute some regulation property. We can call them gene. But we just keep that in mind. It's only based on your definition. Okay. Does anyone know this term, central dogma? You learned it in high school, right? No? Central dogma. Central dogma was the theory which was proposed by Francis Crick. This is Crick. This is Crick. This is Watson. Yeah. After they discovered the structure of DNA, based on the double-strand DNA structure, Crick proposed the central dogma theory. He called it dogma because at that time there was no evidence to support it. It's only based on the DNA structure. But later, in the following decades, more and more evidence support the dogma, the theory. So the central dogma is basically it's a description of the information flow in biology. So from DNA to RNA, then to protein. This part is called transcription. This part is called translation. And the genetic information can also pass through DNA, from DNA to DNA. So this is DNA replication. And nowadays we know that for some viruses, it can also transfer from RNA to DNA. So that's a reverse transcription. So this is basically the central dogma. And this is the key points of modern biology, of molecular biology. There is, someone mentioned that there's some update to you. Watson Crick. Dr. Watson just passed away several days ago. Okay? He wrote a book called Double Helix, and it's a famous book on DNA structure. Dr. Watson was the director of Cold Spring Harbor for a long time, decades. He set up the Cold Spring Harbor. And Cold Spring Harbor is one of the famous biological laboratories in the world. They had a lot of discoveries. When I was in New York, we have collaboration with Cold Spring Harbor. I used to visit their labs. It's not like a modern laboratory. It's like very old building, but they had a lot of discoveries there. And Watson was director there. and he lived just next to the lab. It's like 2 minutes walk from the lab, and they usually had some party in front of his house. And yeah, there are some critics on Watson on his idea, his speech. Yeah, he always say that black people are not as smart as white people, and which is very, yeah, like that. I don't recognize this idea correct, but this, because of this, and the the Cold Spring Harbor finally get rid of his picture from the lab. So that's awesome. But these two guys are very smart. They, based on the X-ray picture, they built out the DNA double-strand structure. That was very clever, and they published their discovery on Nature with just one page, and that gave them Nobel Prize. One page paper. Hopefully, you can do that. Okay, one page paper. Okay, so next we'll introduce the bacterial chromosome. Something wrong with the PPT. This, on my computer, it's just a complete sentence, but I don't know what happened on this computer. The chromosome is, well, the biggest DNA in the cell. For bacterial chromosome, it's a long single molecule, and it's a double-stranded circular DNA. Circular DNA. And usually, in a bacterial cell, there is only one chromosome. One chromosome. But I mean, usually it does not mean it's true for all species of bacteria. For some bacteria, they might have more than one, such as Vibrio cholera. It has more than one chromosome, and usually it's circular DNA. It means that in some bacteria, it might have linear DNA as its chromosome. And bacterial chromosomes are smaller than eukaryotic chromosomes. Like in human chromosome, we have 46 chromosomes. The total length of human chromosome, do you have any idea about that? The length of human DNA. It's on billion base pair level. I think 3 billion base pairs. But for bacteria, it's on million level. Million level. Let me show you an example later. And bacterial chromosome, sometimes they have the operon set up in the DNA. An operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. So, example of this is that you have several genes linking together, A, B, C, D, several genes linking together, head to tail. So when it's transcribed, it transcribes all those genes together to form a big messenger RNA. And the reason for this is that all these 4 genes, A, B, C, D, all these 4 genes might function together. So they are involved in some pathway. Maybe they are encoding 4 different enzymes in the same pathway. So they are linked, they are connected. So they are arranged in the operon. So when it's cracked, all the 4 enzymes will be made at the same time. And also, in bacterial chromosome, it does not, in the bacterial gene, it does not have introns in it. Do you understand the term intron? Like in human gene, our genes are not continuous. So it's cut into pieces, intron, exon, intron, exon. When it's transcribed, there is the RNA splicing to materialize the messenger RNA, to cut the introns out. But for bacteria, there is no intron. All the genes are continuous. After transcription, it can be translated into protein without splicing, RNA splicing. So this is a bacterial chromosome. And another element in the bacterial cell carrying the genetic information is the plasmid. I'll introduce that later. So this is one example of bacterial genome, the bacterial chromosome. This is the E. coli chromosome. And you can see that it's one single molecule, okay? Even though it's very long, here is the length. It's labeled on it, actually. From 0 to here, this is a 4 million base pair. So the total length is about 4 million, but less than 5 million. 4 million base pair in one single molecule. And it's a circular DNA, okay? This is the E. coli chromosome. And this is, 4 million base pair is relatively long. When the DNA is released from the cell, you can see these are the DNA structures. It's much longer than the cell itself. I think I showed you this picture before. So in a live E. coli cell, all those DNA must be well organized together, okay, in the live cell. This is genome. Another element for genetic information is the plasmid. Plasmid is extra chromosomal DNA. So here extra means outside. It's not on the chromosome. And this genetic element is capable of autonomous replication. So it means that a plasmid is a DNA not on the chromosome, okay? It can replicate by itself. So its replication is not influenced, not regulated by the bacterial chromosome, okay? So the properties of plasmid, first, it's autonomous replication. And also plasmids are not essential for the survival of bacteria. This we mentioned that before. If you try to get rid of the plasmid, the bacteria still survive. But the presence of the plasmid might contribute or provide some property for the bacteria. For example, if the plasmid carries some antibiotic resistance gene, and this bacteria will be antibiotic resistant. But if you remove the plasmid, the bacteria still survive, but will lose the resistance property. And also, some plasmid can be transferred from one cell to another through different ways. For example, through conjugation or transformation. And that's a different method for gene horizontal transfer. We'll talk about that in detail later. Here are some plasmid map. This is just a model structure of a plasmid. And this is a real plasmid. For any plasmid, if no matter it's isolated from the nature or constructed in the lab, we usually give them a specific name. And this specific name of a plasmid begins with a little p. So this little p means plasmid. Then there is letter and number. This is the full name of the plasmid. This plasmid, PBR322, the total length is 4,361 base pair. So compared to the chromosome, it's much smaller, right? The chromosome is a million base pair. So this is only 1,000 base pair, much smaller. And on this plasmid, you can find several genes. This gene says AMP, means ampicillin resistant gene. So this plasmid, the bacteria carrying this plasmid will be resistant to ampicillin antibiotic. Okay, so that's just one example. You don't have to remember this detail, okay? Classification of plasmid. Based on their properties, plasmid can be divided into different categories. First, based on the transfer property, some plasmid can be conjugative, which means that they can carry out the conjugation procedure to transfer the plasmid. And if some plasmid cannot carry out conjugation property activity, they are called non-conjugative. And based on their phenotype, and there are fertility plasmid, bacterial plasmid, and also resistant plasmid. Fertility plasmid means that this bacteria carrying this plasmid is able to express the sex pili, and that enables the bacteria to transfer gene from the donor cell to the recipient cell. So that's the fertility plasmid, or for short, we call them F factor. Resistance plasmid means that they carry antibiotic resistance gene. While bacteriocynogenic plasmid means that this plasmid carry the gene to produce bacteriocins. Okay, bacteriocins. One example is the colicin. Bacteriocin is a substance which can kill other bacteria. But this bacteria they kill is something related to them. It's like a relative to them. It's not a broad spectrum antibiotic. So nowadays, we don't use bacteriocin as antibiotic to kill other bacteria because this is not a broad spectrum, and the value of utilization is not very good. Okay, so this is a bacteriocin plasmid. Okay, next I would like to introduce a special structure of the bacterial genome called transposable element. Transposable means what? Transposable means movable, right? Something can move. So that's the segments of DNA which are able to move from one location to another. So this is the important feature in the DNA. Not only in bacteria, even in human genome, in the genome of plants, they carry the transposable element. And this movement look like random movement. It's not a homologous recombination. And the transposable elements are not capable of self-replication. So this is different from plasmid. And this movement or transposition is mediated by site-specific enzyme, recombination enzyme called transposase. And transposase is an enzyme or protein. It is encoded in the element itself. This fragment of DNA carries the enzyme gene. And sometimes the transposition may be accompanied by duplication, which means that this element can make another copy and paste to another location, not jump from one location to another, but copy and paste. There are two types of transposable elements. The very simple type is called insertion sequence. Okay, insertion sequence, this is the most simple transposon, and for short, we call them IS. So insertion sequence. This insertion sequence is first in the middle, it carries the transposase gene. This gene encodes the enzyme for transposition. But on the two end, it carries an inverted repeat. Do you understand an inverted repeat? Let's check the sequence, okay? For this strand of DNA 5' to 3', it says ATCCGGT. Well, on this strand, if you read the sequence, it's ATCCGGT. So this pattern, we call it inverted repeat. Okay, here are some property of insertion sequence. Okay, so that's an element carrying no other gene except those involved in transposition. So it carries only one gene, the transposase gene. And it has the flanking inverted repeat. And this IS is important to make insertional mutation. You can imagine that when this sequence is inserted in the middle of some gene, what will happen? You have a gene encoding some protein, and you have insertion exactly in the middle of the gene. It will disrupt your gene, right? It will inactivate your gene so that it can make some insertional mutation. And with this method, we can actually study the function of this gene. And when this gene is inserted, is mutated, the bacteria might lose some function. The phenotype might change. So this is the first type, very simple transposon, the insertion sequence. But usually, the transposon structure is more complicated because, like this one, this is one IS. So you have two insertion sequence flanking the DNA, while in the middle, it carries more genes. For example, it carries resistance genes. So together, it forms a transposon, the regular transposon. So that's an element that carry other genes, such as the resistance gene, involved in addition to those involved in transposition. That's a transposon. Currently, scientists have discovered a lot of types of transposons. And because it carries a resistance gene, we can use transposon to make antibiotic resistance, to make the bacteria antibiotic resistant. Okay, so let me show you a short video on the transposon. It's like a cartoon showing us the different way how the transposon works. Let me show you.
Transposable elements, which are found in virtually all cells, are segments of DNA that can move from one location in the genome to a target sequence in another. A simple type of transposable element called an insertion sequence or IS element is only 700 to 1500 base pairs long and consists of two basic parts, a coding region for an enzyme called transposase and two short inverted repeat flanking sequences. The cellular machinery transcribes the transposase coding region and translates it into the transposase enzyme. Transposase catalyzes the transposition process. The transposase randomly selects one of many possible target sequences where it will move the insertion element. The IS element, depending on the particular type, may enter the target sequence by one of two general mechanisms: replicative transposition or non-replicative transposition. In replicative transposition, transposase nicks the transposable element, and the nicked ends attack the target sequence, making a staggered cut in the target DNA. DNA polymerase fills in the large gaps, and DNA ligase seals the remaining nicks. A recombination event resolves the two molecules. As a result of replicative transposition, the cell has a copy of the transposable element flanked by duplicated target sequences. Non-replicative transposition is sometimes called cut-and-paste replication. In this process, transposase brings inverted repeats and flanking DNA together. One phosphodiester bond is cleaved on each strand at opposite ends of the IS element, yielding three 3' OH groups. Each 3' OH end attacks the other strand of the double helix, creating a new bond that results in hairpin structures. Host carrier DNA is ejected, and the hairpins on the IS element are re-nicked and attack target DNA. From the 3' hydroxyl groups, the cell's DNA polymerase molecules fill in the gaps, and DNA ligase seals the remaining nicks. In this way, the element jumps from one DNA molecule to another. To summarize, in non-replicative transposition, the transposable element is cut and pasted from one DNA molecule to another. In replicative transposition, the transposable element is copied to yield two transposable elements, one in the original position and one in the target DNA.
Okay, so here are the two different type of transposons. One is that the original copy does not change, but it make another copy at another location of the DNA. So basically, it's like copy and paste. It's like when you write something on your computer, you copy some sentence and paste to another location. Copy and paste. Well, another type is called cut and paste. When you write your paper, you found that, well, this sentence is not okay to put here. I want to move it to another place. So you choose the sentence, cut and paste to another part of your paper. So it's cut and paste. So two different type of transposons. And talking about transposons, actually, it's also related to Cold Spring Harbor lab. A long time ago, I think in 1970s, 1960s to 1980s, there was a female scientist in Cold Spring Harbor Laboratory, and she worked on corn, maize, and she started the transposon in maize. You know, when you eat the corn, you sometimes you would see some seed with different color, right? Like some purple color, deep color, and that is due to transposon. So that scientist worked on that, started transposon. I cannot remember her name, but she got Nobel Prize in 1981, I think. Yeah, also in Cold Spring Harbor. And it was said that Dr. Watson didn't treat her well. So, okay, so that's okay, transposon. Before the end of the lecture, we want to introduce another phenomenon. It's called bacteriophage. So, yeah, bacterial is related to bacteria. What's the meaning of phage? Eat, right? So engulf, eat. So bacteriophage is the virus which can specifically infect bacterial cell as their host. So they are obligate intracellular parasites that multiply inside bacterial cells by using the host machinery. So that's the virus which can only infect some bacteria. So they are obligate bacterial virus. They don't infect human. They don't infect human cells. And for the phage structure, And for a long time, we get familiar with this structure. I think you saw it in your high school textbook. But this is just one type of bacteriophage. Like T4 phage, T2 phage look like this. It's a complicated structure with head and tail, and tail fiber and base plate. And it's so interesting so that this structure is popular. And its composition of the composition of bacteriophage, as long as you know it's a virus, it's the composition of viral particle, nucleic acid and protein. Nucleic acid is their genome, okay? It can be DNA or RNA, but cannot be both, okay? And protein, they use protein as their structure component, such as the capsid coat to protect the protein. And also the tail is also made up by protein molecules. So two major composition, okay? And because it's a virus particle, it's small. It's on the nanometer level. And actually, bacteriophages can be in different structure. This is the most popular one, T4 phage, T2 phage. But it can be in different structure like this T3 phage with a very short tail, while T1 phage with only one long tail and no plate. While this phage is M13 phage, but it's just helical structure. See here, it's a helical structure. So you should have the idea that bacteriophages might look different. And the one we are familiar with is only because it's useful and also it's interesting. And bacteriophages can be classified into two types. One is the lytic phage or the virulent phage, which means that this phage, after infecting the bacterial cell, they can multiply inside the cell, but finally they will lyse the whole cell to release the viral particles. Another form is called lysogenic phage or temperate phage. This phage can lyse the whole cell, but they can also enter the quiescent state. Okay, so they actually integrate their genome with the whole cell DNA. So that's the lysogenic state. Let me show you a short video on this two different type of phage. And after we watch this video, we are done today, okay? I beg your patience.
Hey guys, how you doing? This is a video to show you the difference between the lytic and lysogenic cycles of how bacteriophages can reproduce. Just remember that this process, both these processes, have only to do with bacteriophages, which is viruses that attack bacteria. It doesn't happen in animal viruses like the flu and other things. So basically, to kind of give you a sense of what's going on here, we see that here we have a bacteria. So this is the bacteria right here, and this is the chromosome of the bacteria, and this would be a bacteriophage. This is the virus that attacks the bacteria. So if you kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA, and it has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The basic phage DNA, the genes in the phage DNA directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus. And I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins, and as you can tell here, the phage DNA is going to now code for the production of more viruses, and of course now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. Of course, lytic means to lyse, or means to cut or break open. So this results in the destruction of the bacteria. And that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria. And so what they didn't show here is that the virus would have landed here, would have incorporated its DNA exactly the same as it did in the lytic cycle, but here's now the difference. In the
2 more
lysogenic
minutes,
cycle, you
okay?
see that
2
instead
more
of the
minutes.
DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times. And every time it divides, you can see that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposed to ultraviolet light or some kind of chemicals, a system called phage induction happens. And basically what happens, you'll see here that that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome, or the nuclear DNA material from the virus, incorporates into the actual phage DNA. And is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic, and I hope you enjoyed it.
Okay, that's for today. Yeah, all right. I guess we left here last time. We talked about the two different types of bacteriophages. One is the lytic phage or the virulent phage that can multiply within the bacteria, then it kills the bacterial cell by lysis. And then the second type is the lysogenic phage or the temperate phage. The phage can enter a special stage called the prophage stage by integrating their DNA into the bacterial chromosome. So again, let me show you the life cycle of the two different types of phages. The first one is the lytic phage. Okay. The first step for the phage infection is attachment or adsorption. So that's the phage, okay, binds to the bacterial surface by the receptor molecule on the bacterial cell wall, okay? The receptor recognize molecule on the phage, okay? This step we call adsorption or attachment. The second step, okay, once it attached to the surface, the phage would inject their genome into the bacterial cell. The second step, once the phage particle binds to the host cell surface, it injects its genome inside the host cell. Usually it's a DNA molecule, but sometimes it can be RNA molecule. Okay. This step we call penetration. Penetration means to penetrate DNA or nucleic acid through the cell wall and cell membrane. The third step is the biological synthesis or biosynthesis. So inside the host cell, the phage DNA would replicate. So that's DNA replication or RNA replication. And meanwhile, the nucleic acid would guide the biosynthesis of other molecules required by the new viral particle, such as the protein shell. So different type of protein molecules required for the new viral particle. Then those molecules will assemble to form new viral particle. Once the viral particle is mature, they can be released from the host cell by lysing the host cell. So the host cell is lysed, and the viral particles will be released to infect other host cells. So this is a life cycle of the lytic phage. Okay, and because the virus, the phage, can lyse the host cell, we can use this to count the number of viral particles, the virons. So this is the assay. We call it the plaque forming assay, or short for PFU. So what we do is that we dilute the viral suspension in medium. In this medium, it also contains the host cell, bacterial cell. Then we make a serial dilution of the viral suspension. So in each dilution, the number of viral particles will be different. Then you plate those suspensions on the surface of the dish, the bacterial culture dish. Okay, and if the bacterial suspension, the concentration is so high, you will see a lot of plaques on the surface. The plaques will fuse to each other. So it's not easy to count the number of single plaques. But if the viral concentration is proper, you would see isolated single plaques like this. So in each, you can see those isolated single plaques. Each flag indicates the presence of one viral particle, because the viral particle infects the host cell at that position and multiply and release more viral particles to infect the adjacent cells. So in this area, the host cell will be lysed, killed. So it will form a clear plaque, a clear halo. So you can count the number of the clear halo or clear plaque. You can decide the actual number of viral particles. This is a very smart method because the viral particle is so small, it's on nanometer level. We cannot see it with a light microscope. And it's not easy to count with electron microscope. But with this method, the plaque forming unit assay, we can actually count the actual number of viral particles. Okay, so it's a very useful method, the assay for the lytic phage. Okay, the second type of phage is the lysogenic phage or temperate phage. And we call it temperate because once they infect the host cell, probably they are not lysing the host cell immediately. They are able to enter a non-lytic prophage state. So which means that they integrate their genome DNA into the host DNA, the host chromosome, okay, and replicate with the host chromosome. So this stage we call prophage. The phage DNA integrated into the bacterial DNA. When the bacterial cells replicate, the phage DNA will also replicate with the bacterial DNA. It does not kill the bacterial cell immediately. Okay? So that's the lysogenic stage or lysogenic phage. And those bacteria carrying the prophages are called lysogenic because a physiologic signal can trigger a lytic cycle. So once it's induced, those bacterial cells can further enter the lytic cycle, and that will result in death of the whole cell and also liberation or release of many copies of the phage. Let me show you the life cycle. So this is a lysogenic life cycle. The phage infect this cell, okay, attachment, then penetration to release its nucleic acid into the host cell. But here, it's special here. The viral genome will form a double-strand DNA, integrate itself into the bacterial chromosome, okay? And when this bacteria grow and replicate, this DNA can replicate with the bacterial cell. But once induced, this DNA can be taken out, okay, enter the lytic cycle. Okay, so this is and then last step is release, also lysing the host cell. So this is special because it can enter a special state called prophage state. In the prophage state, the phage DNA would replicate together with the host bacterial DNA. Okay, because of these two different life cycle, and they can cause different effects. Later when we introduce transduction, and only the lysogenic phage can carry out a specialized transduction. We'll learn that later today. Okay, so this is a lysogenic phage, and there is another term we want to understand is called lysogenic conversion. When a temperate phage may induce a change in the phenotype of the infected bacterial host. Okay, and this phenotype change we call lysogenic conversion. And this is not part of the urophage cycle. Okay, in this way, what happens is that if the phage genome, the phage DNA, carries some special gene, such as antibiotic resistance gene, virulence factor gene, okay, when it integrates its DNA into the bacterial chromosome to form the prophage, that special gene is already inserted into the bacterial chromosome. So it can be expressed to enable this bacteria like antibiotic resistant, or that this bacteria can be virulent to cause infection in humans. So that in this way, a temperate phage can play a role in the spread of virulence factors such as the genes of exotoxin or exoenzymes, or maybe like antibiotic resistance gene among bacteria. So this is a very important way for gene transfer, lysogenic conversion, so that the bacteria carrying the prophage might have a special property. And it's interesting that a lot of human pathogens, only when they carry some prophage, they are virulent. They can cause infection in humans. But without the prophage, they are not. We'll learn that later. I'll show you a short video talking about the problem. Okay, the phage, is especially the lytic phage, it can kill the bacterial host. Okay, when it infects the bacteria, it kills the bacteria. So there's the idea that can we use a phage to kill the bacterial pathogen so that we can cure bacterial infection? Is that possible? Yes, it is possible. Actually, this is not a new idea. This idea was proposed a long time ago, maybe over 100 years ago. When people discovered bacteriophage, they thought about this idea. Well, we can use phages to kill bacterial pathogen. But later, because of the discovery of antibiotics, and antibiotics are so specific, and we can easily use antibiotics. And this idea was put aside. So for a long time, scientists were not working on the phage therapy. But in recent years, we know the problem. Because of long-term utilization of antibiotics, we give the environment some pressure so that the bacteria will evolve and become more and more resistant. So the current antibiotics cannot work anymore, and we cannot find new antibiotics easily. So people came back to this idea of phage therapy. Maybe we can find some phage to fight against the resistant strains. Okay, but right now, more and more labs are working on this, but it's not easy because usually you need to use a cocktail of phages. One specific virus is not enough, so you need several phages to work together. And also, there is another problem. If you utilize phage to kill bacteria in our body, the phage themselves are also antigens from outside, not your part of your body. So your immune system will kill the phage. Your immune cells will recognize the phage and kill them. So it's not easy to carry out. There is one famous example of phage therapy. So they use phages to kill a bacteria. This is a real story happened about 10 years ago. In 2015, in America, in UCSD, University of California, San Diego, a professor of that university got infected by a special antibiotic-resistant bacteria. And they tried all the antibiotics at that moment, but cannot cure him. But luckily, his wife, Stephanie, worked on global health, and she knows a lot of scientists working on phages. So they tried this alternative method to cure her husband. And finally, yeah, her husband survived. Let me show you this video on this phage therapy. Okay.
They were having a great time. They'd seen the pyramids, and they were on a boat headed up to Luxor. And as the boat was pulling into Luxor, Stephanie texted me and said that Tom had developed abdominal pain and was unable to eat, and she was worried that he developed food poisoning. She then texted me back and said, the pain is going through to his back. Could this be pancreatitis? What should I do? And I said, you need to get into a hospital. The last thing you need is pancreatitis in the middle of Egypt. So an organism we have a lot of respect for now. So after a little over a week in Germany, Stephanie was able to work with people here to get him flown to UCSD. And he was hospitalized here at UC San Diego just before Christmas.
I wasn't ready to let you go. And I came to you and I held your hand and I said, honey, they're doing everything that they can do. There's nothing that can kill this bug, so if you want to fight, you need to fight. but do you want me to kind of pursue some alternative therapies? And we'll leave no stone unturned. Around that time, a colleague of ours who's at UCSF said that a friend of hers had been treated with phage therapy and flew to Tbilisi, Georgia, where she had this miraculous cure. And I thought, wow, that sounds kind of far out. But wait, I remembered learning about phages when I was an undergrad at the University of Toronto. So I thought, wow, and I looked it up and I saw that it wasn't licensed and it certainly wasn't FDA approved, yet there were some papers saying that this was a promising area for the future. So I wrote Chip Schooley an email and I said, what do you think about phage therapy? And he said, what an interesting and intriguing idea. If you can help identify some phages that are going to be reactive against his isolate, I'm going to give it a whirl. We set on a phage hunt. Took just a couple weeks, but an international effort of researchers and people at the Navy Medical Research Unit came together to make this happen. We couldn't have done it without the UCSD team.
What they literally mean are bacteria, phage. They eat bacteria. These are small viruses that are ubiquitous. We're loaded with bacteriophage, and on any given day there's a dance going on in our GI tracts of bacteriophage attacking bacteria. In the early part of the 20th century, there were a lot of efforts in France and what became the former Soviet Union to use these organisms as therapeutic agents in people with difficult to treat infections. Toward the late 30s and early 40s, there was less interest in this in the West because antibiotics began to emerge, and people in the West thought antibiotics would be able to take care of our problems with bacterial infection. There had not been a lot of experience with treating people with bacteriophage, and almost none giving it intravenously in the U.S. So we relied on some of the literature from the Soviet Union, but a lot of it was really being worked out kind of on a first patient basis as we went, using a combination of the previous literature, our own intuition about how these phage would circulate, and advice from people who'd been thinking about it for a long time. Within about 48 hours, he woke up and turned to his daughter and said, "I love you."
Had there been phages available in the course of my illness when I first got ill, I wouldn't have been nearly as sick as it turned out I was.
There's a lot of interest in bacteriophage therapy. It's been gradually emerging over the last year or two as efforts to develop more traditional antibiotics have not really made the progress people had hoped. Bacteriophage have been kind of brought back off the shelf as a potential new approach to therapy. They're not simple to use. You have to develop a cocktail for each patient's own isolate. They seem to be relatively safe to give, but they're going to be difficult to develop from both the research perspective and also from the regulatory perspective. Now the good news is we have awfully good tools now to do this, robotics and much more sophisticated molecular tools that enable this to be done. 10 or 15 years ago it would have been impossible to contemplate doing this. And so I think, although there's a lot of research to be done, I think there are going to be a lot of clinical applications where this approach to therapy may be very beneficial to patients.
You can't emphasize enough how much, I believe, that phages really can change how we treat people in an economical way. So I see the third world and other places benefiting as much as we do. And in my case, I can't tell you how much it costs to take care of her, much more than, I would like to believe. But if this can be done cheaply and really low-tech way, we can save millions of lives. And so it's, I think, the future of medicine.
Okay. So even this is just one single case of phage therapy, and it's still possible that it's worth it to try this method. And nowadays, there are more and more clinical trials registered for phage therapy, and especially for this one in this institute, UCSD, they have a couple of clinical trials registered to use phage to cure staphylococcus infection, as I know. And as I said, it's not easy. And the first thing is that you need to have a library of phages, a lot of phages to try in a short time. And a couple of years ago, in my lab, we spent a couple of years, like a year and a half, to find new phages from the environment. We want to isolate new phages against staphylococcus, like MRSA. But unluckily, we didn't find any new phage from the environment. Because, if you learn better and deeper on microbiology, you will know that actually most bacteria are resistant to phage infection. They have a system called CRISPR system. CRISPR system is an immune system which can prevent phage infection. So, well, we spent some time on it, but we didn't find new phages. But there are some institutes in a former republic country of former Soviet Union. They have an institute or some center. They have a big collection of different type of phages. So nowadays, a lot of people went to that center as their last hope to cure their infection. So that's what's that country's name? Georgia? Is that? Yeah, Republic or it's not far from Ukraine, right? Yeah, that country. They have a center on phage therapy. They have a big collection. Yeah. Okay, let's move on. Let's back to our phage.